Uh, just one moment. No? Like my screen got moved actually in the process. So once again, please. Okay. So we have our. So we don't have the second speaker here, Professor Saurav Raghuvanshi. So in that place, Professor Ramachandran is talk is moved forward, and so it's my pleasure to invite Professor Ramachandran for this particular conference, and also consented to give a talk. I know Professor Ramachandran for quite a bit of time, and let me actually give you his brief background. Like he is basically a chief scientist, and he is a professor of ACSIR in the Faculty of Biological Sciences. And his wide interest is basically the disease comorbidities and small molecule inhibitors against tuberculosis. And his expertise is also in integrative bioinformatics and molecular biology, data analytics, developing packages in all. In fact, I have used a couple of his packages actually. And also that he has a lot of experience. He has more than 25 years of experience, and post PhD includes more than 14 years teaching experience in bioinformatics. And he is also leading in IGIB skill development programs in genomics and molecular technologies for industrial applications. And also to reskill programs in R and literature to pathways data mining using text mining. His recent achievements are basically he has developed new methods for prediction of virulence factor based on machine learning, integrative immunoinformatics, PubMed text mining, gene atlas for type 2 diabetes comorbidities. His not only his work, his group work focuses on bioinformatics of disease comorbidities and identifying new inhibition strategies as developing alternatives for tackling AMR. So I request Professor Ramachandran to take the floor. And if you do have any questions, you can either use the chat box, or you can interrupt, or you can ask the questions at the end of the lecture. Okay, Professor Ramachandran. So I can see your screen actually. Your voice is muted actually. Yeah.
Yeah, my slide?
Yeah, I can see your slide. You can make it a little bit big actually.
Yes, yes. Okay, now?
Yeah, absolutely fine.
Right.
Thanks.
So I'm going to speak to you about our work in the last many years on pathogen bioinformatics and disease comorbidities. So thank you, Raghav, Sriram, and everybody for giving me an opportunity to share our work with you. So I'll start with more than, I would say, more than 20 years old observation, and how this observation actually led to many of the projects that we did. So this is the sequence of a protein from a nematode pathogenic worm, which is called Strongyloides stercoralis. And it is, what we found was that it is a very potent antigen. So if you look at the sequence of this protein, it is highly charged. It is having biased amino acid composition. And you can see the amino acid residue glycine is repeated, not every third, but at multiple of 3. Sometimes it is every third, like here. Very strange protein. So I was very much struck by this particular amino acid sequence and its very high antigenicity, because in the serum of patients infected with this worm, they had very high titer of antibodies against this protein. Later, when genome sequences started coming, then because of this observation at the back of my mind, I wanted to see whether such type of biased amino acid composition proteins exists in different genomes. This was the early times when Methanococcus januschi, Haemophilus influenzae, and many other of these proteins were, where sequences were appearing. So you can see that in a plot of amino acid composition distance, which is normalized to the length of the protein, and you plot it against the percent hydrophobicity of the protein, then you can see that in a very narrow window of hydrophobicity, most of the proteins are encoded in the genome. But when you look at the amino acid sequence composition, then you can see that the amino acid composition is having much bias, and there are outlier proteins in this genome. So these are outlier proteins which are at the top away from the cluster. So not only that, you can also see that there are two types of clusters seems to be forming, and most of the proteins are really clustered very densely in a narrow range of amino acid composition makeup. And then there are few which are separated from this. And then there are few which are totally away from the main cluster. So we were struck by this again, that such amino acid compositional biases are there in other pathogens also. And we then looked at tuberculosis, and you can see in the bottom plot here, here the hydrophobicity, there is some wider range compared to Haemophilus influenzae, but then there are proteins which are having very high biased amino acid composition. Then we looked at these proteins and found that some of them are a membrane protein, part of integral membrane protein, or circumsporozoite antigen, or surface proteins, or permease, or proteins involved in virulence, or virulence in tuberculosis also, and or proteins involved in adhesion, that is binding of the pathogen to the host, then cytoadherence accessory protein, and so on. So we then developed a method to look at this uniformly from all the pathogens, and that work was published later. So it told us that there is a common thread of compositional preferences for the proteins in pathogens, and maybe it's there in other organisms also, but our focus was on pathogens. And then there seems to be some kind of groups of proteins which have been present in these genomes. So this was a revelation, very important revelation from the genome sequence work in the early days at that time. We then developed some algorithms to look at this more objectively and with in a systematic way. And then we looked at many genomes and prokaryotic genomes and found that what happens is there are some proteins which are evolving in different species. And then there are some pathogens where you can see this by this arrow that there are simple duplications resulting in small sized different families. Then there are some families which are taking birth from large duplications. Okay, there are some families which are taking birth in large duplications. So this showed that there are different modes of evolution in the pathogen genomes to acquire proteins of biased amino acid composition and then duplicate it and then change it and use it for different purposes. So there are three levels of dynamics of gene expression in the pathogens. What we learned from all this work summarized in this slide. There are some constant genes, they will always be there. And they introduce what is called constant expression, not much variation among the different strains. Then there are constant genes which undergo variable expression. That is because when there are conditions are changed, like they are in an outside environment, then when they go inside the host, then certain changes happen which were not being expressed before, and then that actually gives some advantages for them to colonize in the host. Then there are variable genes which have either regular expression or they may be varying also. Some people even call it variable antigens. And this type of variable antigens are also part of the gene expression of these pathogens, by which they try to fool the immune system of the host and keep going in its survival. Now, while going through this work, I was very excited because of my initial observation and then going through all this genome analysis during my early days of getting a job here, and students also opening the gates by doing marvelous work. My excitement knew no bounds. I didn't care for anything in life. Then I found that lo and behold, there were other observations people had made. Some Japanese papers had come, then some European papers also had come. What they were looking at was very fundamental properties of proteins. And one of that fundamental property is its location. A given protein has a function for its location. So its location being important, the location is information for its location is embedded in its own amino acid sequence. What a marvelous creation. So we are talking about multi-layer information here present in the protein sequence. So it involves amino acid composition, and it also involves arrangement of amino acids, whereby where the protein will go is already predetermined, and whatever machineries are there in the cell they will help in ferrying that protein to that particular location. So that the protein once made, it doesn't wander about here and there, searching for some location, not like an orphan, but it will go through a guided mechanism to its destined location, and there it will do its function. So the observations made by these people were broadly categorized like this, that the intracellular proteins and the extracellular proteins, they were very different in amino acid compositions. And then there were many reasons explained for that, because the extracellular environment is more harsh and the protein has to withstand all that harsh environment and therefore maintain its structure and integrity. Whereas the intracellular environment is more conducive, helpful, goody environment, and therefore its amino acid composition is different, and so on. So what we were looking at from another angle through accidental observation, and what other people were studying now started to come together, and our excitement kept on increasing. Then we used this principle straight away to encode it through artificial neural network, whereby we trained the network using a set of proteins that are virulence factors, known virulence factors, and their sequence amino acid composition, and other proteins which have no chance of being virulence factor. They are basically enzymes and they will be inside the cell. So they have a different amino acid composition. So we had the positive and the negative data set, and we carried out this training, and we were able to successfully develop an algorithm for identifying virulence factors. It is still regarded in the literature as a time-honored algorithm. It has not been beaten yet. People are trying to do it, but they have not so far become successful. So it is still very widely used in the literature. Then we asked the question that we pushed further. We wanted to see whether our excitement can continue to other pathogens. So we more or less covered the bacterial pathogens. Now we wanted to cover the malaria parasite. But parasite is in the eukaryotic domain, and fundamentally that evolutionary kingdom is very different from the prokaryotic one. So initially, although we were excited, we also were a bit hesitant. So I somehow Dr. Vidyasagar and Yogeshwar Rao, they believed in me, and they gave me some funding and said, okay, Ram, go and do this work. So we successfully finally developed an algorithm, but this time based on support vector machine, which I went to Raghav's lab in Chandigarh and learned like a student from his student. And then came back and we could develop it successfully, which helped us in identifying the virulence factors of Plasmodium in different stages, because this parasite undergoes different stages during its life cycle of infection. It is also highly cited, and there is no parallel algorithm developed yet for this. Then later, we found that we can look at the fungal pathogens, because the fungi is a very diverse kingdom of eukaryotes. And these pathogens, they give a hard time in clinical treatment. Although some drugs are available, but still a fungal infection is worrisome. And nowadays, because of the mucormycosis scare, that has happened in the second wave of COVID, the fungal infection has really come as a big demon in our daily life. So at that time, this was not so important. In fact, people were treating it as only opportunistic infections. So we were basically developing algorithm for identification of virulence factors, which can be used potentially for developing antifungal vaccines for at-risk people. And here also we were successful in developing using support vector machines and amino acid compositional properties of the proteins of the virulence factors, which were very different from the other proteins. So we took advantage of that and were able to develop these algorithms with high prediction Matthews correlation coefficient. What we saw was happening around at the same time was that Raghav was developing many immunoinformatics web servers, one after another. B cell epitope prediction, then like that. And we thought that we should integrate. This is the time to integrate and develop methods which can help in design of vaccines, especially what they call subunit vaccines at a faster level. It was also at the time when there was confidence developed in the genomics community because of the successful development of meningococcal vaccine using reverse vaccinology approach, where they went from genome sequence, computer analysis, and then developed the vaccine. So all these developments came together and they were influential in our development of immunoinformatics included in this pathogen. So this particular paper has both virulence prediction as well as immunoinformatics integrated together. As I told you that Raghav was developing this immunoinformatics portals, and then other people like Darren Flower and others were also developing algorithms for immunoinformatics from other countries. So we could develop this server. Map was already developed thanks to the funding from some people, some help from Sagar and Dr. Rao, and then we could integrate it here with immunoinformatics servers to produce Malvac, which was published and still being used and cited. And this is for the fungal RV, where once again, using different criteria, one can shortlist the proteins and go for vaccine testing. Then a little later, what happened was we went back to that mycobacterium pathogens. Now, by this time, what happened was lots of genome sequences of many mycobacteria strains were now coming into the genomics databases. So we thought this is a very good opportunity to use the different virulence factor predictions as well as the algorithms from immunoinformatics together and build up this data for the community to take advantage of. So it's basically, in a way, you analyze and sort it and make the data available for the public, which is already from the open data only. And it can help the people who are interested in developing next generation vaccines for these pathogens, deadly pathogens. So this is for the mycobacterial strains in the R platform as well as in the web server, which is available, and it can be used for this purpose of developing strategies, anti-infective strategies. Now, we found that in the recent COVID times, this mucormycosis has started. It has become a very big demon, as I referred to earlier, suddenly. So we then developed a pipeline for this mucormycosis pathogens. And in this case, we actually started from the UniProt. UniProt, by the way, has become a fantastic database with beautiful integration of all data from everywhere. They have really done a fantastic job. And from there, we could get all the proteins from different pathogens causing the mucormycosis disease. And then from there, we developed the pipeline, and we now have given that data for public to use it for developing anti-mucormycosis for at-risk people. Now, as today morning, the pathogen problem is not only this problem. The pathogen problem is a multi-dimensional problem. So in the morning, Harpreet sir had given a beautiful overview on AMR, antimicrobial resistance, and how very little work has been done in that and how much work remains to be done. We also felt that while going into that. So we wanted to look at this problem from a different angle. And it was basically, an extension of our work on trying to develop small inhibitors against tuberculosis. So as an extension of that, we wanted to look at this work. And as you can see, AMR will become a major killer in about 2050 if we don't do something about it now. So people have realized this. Many people have realized this all over the world. And therefore, what they are doing very innovative strategies so that the AMR does not bounce back. So that is very remarkable. We were very excited by looking at the strategies what people have developed in this area. So a lot of people have developed the problem side of the informatics tools, which includes CARD, ARDB, MegaRes, whereby if you have a genome sequence, very quickly you will get to know what are the antibiotic resistance genes present in this genome or in the sample. And that gives basically an idea for the clinician as well as the team of doctors that how to go about the treatment for this particular case. The solution side, we then found out that Raghav has been at the forefront, which is welcome development, focusing on antiviral peptides, anti-tubercular peptides, and so on. Basically, anti-peptide kind of strategy. New peptides which will kill the pathogens. So these are beautiful resources developed by their group. And then there is another group in England who developed antibiotic DB on looking at what are the other compounds or drugs that are undergoing, compounds that are undergoing clinical trials in various different phases, what are withdrawn and so on and so forth, which gives a very quick overview of the developments in this field. We were interested to know what else is happening. And in order to do that, we found a very different approach. So the antipeptide approach and the antibiotic approach is basically a entity-based, known entity-based approach. I mean, you know that what you're looking for, and then you search for that from the literature, what work has been done and what is most useful. But when you don't know what the entities are, then we don't know how to go about this work. So my students, they developed this sentiment-focused work where the authors are developing a particular strategy and expressing their sentiments, how it will work against the big Himalayan mountain type of problem of AMR. So you can see the sentiments that is being expressed for that. And using the sentiment vocabulary, we then did the search. This has the advantage that I don't need to know what entity it is. It can be any entity. It can be a biological, it can be a chemical, it can be a nanoparticle, it can be anything. But the sentiment is important here. So using this strategy and this text mining package which we developed, which will be described by my student in 1 or 2 days, we came to this set of vocabulary which actually expresses the kind of therapeutics that people are trying to develop. And they are trying to develop in such a way that AMR doesn't come in those new therapies. Why? Because AMR basically comes because you are having single target therapy. If you use a multi-target therapy, then there is very less chance for AMR to come. This is one of the thought process that is prevalent in this particular strategy. Now, using the sentiment approach, we got the new approaches that people are developing, which includes biological agents, nanocomposites, etc. But we also have to get the objective data, that is how good that particular therapeutic development is, strategy is. So for that, we have to get objective data. So sentimental vocabulary gives subjective data. For objective data, you need a different vocabulary. And using this vocabulary, you can find that the data does not express any sentiment. That particular data does not express any sentiment where this vocabulary words is there. That's what it should be. That's what science is. It should be neutral completely. And then people should be left to decide how to do the assessment and how to go about the work. So by combining the two approaches, we have been able to develop all the therapeutics that have been identified so far, phage therapies including. And phage therapy, I'm mentioning that it is not approved yet, but in a wide scale, but on very critical situations, it was approved and some people's lives were saved using phage therapy. Then there are so many other therapies like adjunct therapy, synergistic therapy, probiotic therapies, photodynamic therapies, and so on, which are developing. So this armamentarium is now being filled, and in order so that we don't have to lose a night's sleep of pending AMR coming and attacking us, we have good hope. So this has been developed into the combat database, and we covered bacteria, virus, fungi, and parasites, all sorts of pathogens. This is available, and the preprint is also available in the as mentioned in this slide. And this resource is available for anyone to use it. It was quite an exciting venture to get into, to get the data for this AMR. Now, in India, it is also becoming the capital of diabetes. So, although AMR is there, we have a lot of problems. We have a lot of health problems. We have infectious diseases, so many things we have. And then we also have this diabetes coming up. And it has become a major health problem, next only to China. And we can see that this is such a disease that it leads to many, many other diseases, which are called comorbidities, such as cardiovascular retinopathy, heart disease, COVID also. Recently, COVID also has come, and this comorbidity word has become very important. Every TV interview, they mention comorbidities in a COVID, during COVID time. So it has become very important, but we've started work from before, so it has become useful nowadays. So how do comorbidities arise? So you can have the risk factors which are leading to different diseases, such as smoke and alcohol consumption. And if both risk factors are there, then you have comorbidities of both the diseases coming. Sometimes a given risk factor, such as smoking, can introduce two diseases, heart disease and lung cancer. Age is also a factor. So given this situation, the interest has speeded up in looking at comorbidities in different countries, including China. And literature is building up on how this is happening and what are the genes involved and how we can target them. So we had prepared this, this is published 4 years ago though. So, but we have updated it. We are keeping on updating it thanks to the students who come and do their dissertations with us. And this has allowed us to keep the database up to date, because nowadays, if you're not up to date, then it is the half-life of use of the database will go down very fast. So either we need funding, and in our fund crunch situation, then you need to depend on students' dissertations for degrees, which they have to do as part of their curriculum. So this actually gives a very comprehensive overview for diabetes and their comorbidity diseases, including tuberculosis, dyslipidemia, and so on. Now, we did not understand how this comorbidity works. So we had to struggle for quite some time. And then we developed this protocol. Looks a bit complicated, going down to many, many different types of data sets. But the essence is that to look at the genes which are differentially expressed at high statistical significance, and then then extract the network data from string and pathways from there. But that gives only static picture. It does not give the dynamic picture. So for getting the dynamic picture, we need to do simulations. And for this, we have used one particular simulation software that worked with us, developed in Israel. That seems to work smoothly. So we have been able to do that and get some insights into how these comorbidities develop. So one view in this comorbidity development is, I'm sorry, can I ask Sriram how much time I have?
So
Hello?
we'll be having 15 more minutes.
Okay, thank you.
Yeah.
So you can see here in this Venn diagram, by merging different genes that are associated with different diseases, what we found was that there are 13 genes at the center. They are mentioned here for which are central to all the different comorbidities, diabetes and kidney, diabetes and eye, diabetes and heart, and so on. Now at the periphery, you can see that there are several genes that are unique to the different disease complications. So the distinctivity is maintained, but then there are certain common pathways by which the different different disease afflictions can develop. So if you don't control diabetes, then these other comorbidities would develop, but this even a doctor would tell you. So what do we gain from this analysis? What we gain from this analysis is that we understand at the gene level that the moment diabetes has started, the risk for diabetes associated diseases have also started. That is the thing. It is not that they are to come later. We can worry later. Not like that. They are also starting because those genes are, some of those genes are also involved, and they are in central pathways with a high degree connected nodes, and therefore it is likely that if you don't control the index disease, which is diabetes, then the comorbidities development will become quite certain. Once the comorbidities are developed, then it is very difficult to reverse. That is the problem. And then it leads to finito. Now, if you look at the different populations, so Indian population data, Chinese population, European and Japanese population, how are they similar or how are they different? Okay, this is a question we can ask. And we found that the connections between nephropathy and the risk factors, common risk factors, stress here means oxidative stress, not the mental stress. Oxidative stress, obesity, inflammation, and diet, they are strongly connected to each other. So when the diet prescription is given, it is imperative to follow that very strictly. That is because the diet is very nicely connected to all these different diseases, and the connections are pretty strong. It cannot be avoided. The other parts, such as inflammation and obesity, are something that one has to take control, and stress and other things. So they all can be controlled with different types of diets. It's not that they cannot be controlled, and some medication is also required. Then some physical activity, prescribed physical activity, compulsory physical activity, all that are also required. So one of the activity I was trying to think was that if I needed something, then I will go to that person by walking instead of calling that person. In my stage, I can easily call anybody to me and they will come running to me. But instead, I go to them. So that gives me some exercise. So it's a different way of lifestyle changing that can help in avoiding these comorbidities. Now, in the case of tuberculosis, how diabetes is making a person susceptible to tuberculosis, we wanted to investigate that, and we found something very interesting. So there's a lot of literature data on which genes are upregulated, which genes are downregulated in the diabetes with tuberculosis patients. But that doesn't tell us somehow that a given person who is having diabetes, is that person going to be susceptible to tuberculosis, and how? So we then try to look at the genes which are involved, host genes which are involved in rendering susceptibility to tuberculosis, and the literature data that people have found on genes that are upregulated in diabetic patients with tuberculosis, and see whether the signatures are matching. Unfortunately, we didn't find much match between a person who is diabetic but not having tuberculosis and people who are having diabetes and tuberculosis. It's not matching, except two genes. One gene is the TACO gene, which is coronin gene, which is involved directly in the susceptibility of helping the mycobacteria to colonize in the host. But the other gene, which is the innate immunity gene, which is defensin A, is also upregulated in both cases. Only thing is, if the person did not have tuberculosis, then the upregulation value is very low, compared to the person who is having tuberculosis, where the upregulation value is very high. Other than that, the signatures, as you can see in these arrows, they are not very much matching. So then in that case, how do we investigate that what is giving susceptibility? Okay, coro1A is there, but is that the only gene that can we can pinpoint to that susceptibility? So we then did that network-based approach and then got the pathways, and then using the gene expression data from the diabetic patients and compared with normals, we then carried out the simulation, and then after distilling all this data into one figure, we found that the necroptosis pathway seems to be highly perturbed in the diabetic patients. Then we searched in the literature, what about this necroptosis pathway? So necroptosis is a necrotic pathway in the apoptosis mediated way. So it's called necroptosis pathway. And it has its own genes, which are shown here in the pathway. And all these genes in the necroptosis pathway is perturbed. And we found there's a recent paper, not very old, that the necroptosis actually helps the TB bacteria to colonize in the host, in the pulmonary areas. So that seemed to be explaining that how this can happen. And if that is so, then we can think of drugs which can try to, adjust this pathway in such a way so as to reduce that perturbed level back to normalcy, like a normal person, and thereby reducing the susceptibility to tuberculosis. This has become a major problem, especially it became striking when the East Europeans migrated to the West. And it is definitely going to be a problem, I think, in our country also, because we are having both the disease in high proportions. COVID, enter COVID. And we find that the comorbidities are helping in establishing and colonization of the COVID virus. And we saw, while comparing the gene expression patterns between many different diseases, cancers, and COPD, type 2 diabetes, asthma, etc., we found that the cancers are the people who are highly susceptible, and then the people susceptible because the gene expression pattern is very similar to the pattern of COVID patients, and therefore it is possible that the viral survival is very conducive in that environment, which is different from the normal healthy person. So given this situation, the NAFLD, that is non-alcoholic fatty liver disease patients and cancer patients, have to be ultra careful against COVID, and other people, they are less susceptible, but to a degree, but they also have to take precautions. Then simultaneously we were looking at another problem which was reported in Chinese literature as well as in some of other literature also. how diabetes and dyslipidemia leads to psoriasis, hyperproliferation of keratinocytes in some places of the body. And so here again, we did the literature mining. We got the genes that are involved in psoriasis, T2D and dyslipidemia. And then we took a common set of genes. And these are the common set of genes which are listed at the bottom here. And you can see the ACE is there, along with ADPOQ and many other genes, fatty acid pathway genes, VGFA, which was there in the diabetes also. Here also it is there. So we then wanted to look at that how this dynamics is going to happen. Basically, the main thing is once an index disease set up, then how susceptible you are and when it can happen to that person to contract the other disease, get the other disease. And once there are two diseases, then you can know that person is in a bad shape. So we found that in the psoriasis differential expression data, we found that all these genes which are implicated in the psoriasis complication and as well as other disease complications, they are differentially expressed only in the skin samples of the psoriasis. So something happens in the skin, but not in the blood. So if you measure in the blood, you won't get anything, it will look normal. But if you measure in the skin, then you get this signal. So this is another new revelation to us that what is happening in the skin is not seen in the blood yet, but it is happening quite separately and somewhat localized in that organ, which is different from what is happening in the blood. And this difference, we put very high statistical cutoff in order to check so that we don't want statistical insignificant data to look at this. And we find that several genes, including the hunger gene, that is leptin gene, the ACE gene, the ADPOQ gene, and the fatty obesity gene, the SART1, VGFA is all involved, and some cytokines are all involved in this. So now we have to do the simulations and try to pinpoint how this pathway is getting disturbed and thereby provide a window to look at how to treat these people so that such a disease cannot arise. So finally, I would like to end my talk here with one quote that is very recent from the Twitter from Professor Vijay Raghavan, who says that there are some scientists who, when they interact with young people, are inspirational. Not because they are great or famous or Nobel laureates, not because they communicate well. This also was a new learning from me, because I used to think many scientists, they are very successful with young people because they communicate very well. But here he says, no, that is also not the case. He says it is because they are often decent and kind and have not lost their childlike enthusiasm. So this struck me. And what I have been recently doing in my activity is to do the show the skills and hands-on work with the students, and then leave in between, so that forces them to think. I don't want to bias them with my thoughts. So let their original thoughts come out and then enjoy the childlike enthusiasm. So that's going to be more of my future occupancy. And finally, I leave with this quote that is from an Indian sage from very old time of Alexander's time. I recently got to know that unless you strip your conditioned mind because there are too many conditioning factors, school, college, exam, this, that. How do you expect to have, hold, or create anything original? So this is most important. In spite of the conditioning, we have to come out and think of original solutions like the AMR solution, like the disease comorbidity solution, like that. Thank you.